heredity and evolution. Hereditary changes, transcription, translation and translocation. Which component of cellular nucleus of living organism carries hereditary characters? Chromosomes, they carry the hereditary characters. Chromosomes are present in the nucleus, and the set called genes are part of those chromosomes. Means a single chromosome can contain thousands of genes. So it's a segment of the chromosome which stores the information about a particular protein. And this is called heredity. That means the transfer of biological characters from one generation to the other. That means the information about the structure and function of that organism is transmitted from the parent to offspring via these chromosomes. Now this was discovered by Johann Gregor Mendel. He did the experiments of monohybrid cross and dihybrid cross in 1886, and that was the beginning of modern genetics. So this Mendel is the pioneer of the modern genetics. It took a long time for him to understand the conclusions. It was about mathematics, how the characters are transferred from the parent to the offsprings and what are the rules that govern this transfer. So that was the beginning of modern genetics. But Mendel didn't know anything about the biological factors which are responsible for this transfer of characteristics. In 1901, something new was discovered, and that was mutational theory. That means even though the characters are transferred from one generation to other, sometimes it happens that there are some changes in the offspring. And these changes are due to mutations, and this was discovered by Hugo de Rice. In 1902, Walter and Sutton, they found the paired chromosome for the first time in the grasshopper cells. So this was the discovery how these biological characters are transferred from generation to generation. And this was due to the chromosomes, and they were discovered by Walter and Sutton. Research started in this direction, but it took a long time. It took invention of electronic microscope when in 1944 Oswald Avery, Maclean McCarthy and Colin MacLeod, they proved that all living organisms have DNA. 1961, the structure of DNA was known. The French scientists Francois Jacob and Jacques Monod, they proposed a model for DNA of the bacterial cells, and then the DNA was known. So you can see that from 1886, the external characters were studied. 1901, mutational theory was developed. 1902, the paired chromosome were observed for the first time. 1944, the DNA was confirmed, means the DNA as a genetic material was discovered. 1961, structure of DNA was known. So the science of heredity, and then it is used in the diagnosis, treatment, prevention of hereditary disorder, diseases in animals and plants. Hybrid varieties of animals and plants could be made. Various industrial processes developed in this genetic engineering. The basis of the expression of the characters in the living organism is transcription, translation and translocation. These are the three main processes taking place in all living things. With the help of RNA in DNA participate in functioning of cell. So what do you mean by functioning of cell? Functioning of cell means the biochemical reactions taking place in the cell. And these biochemical reactions are responsible for the life of that cell and of that organism. So the control of structure and function of the body is done through this. Information about protein synthesis is stored in DNA, as we have seen that the chromosome, they contain the chains and the part of the chains named genes, which store the information about a particular protein. But how this information, using this information, how the proteins are made? These proteins are synthesized by DNA through RNA, ribonucleic acids. This is called central dogma. So central dogma is preparation of proteins required for the life of that cell and organism, which are synthesized by DNA. That means information is stored in DNA through RNA, and three kinds of RNA used in this. First is mRNA. The first character in this entire process is mRNA. mRNA is produced as per the sequence of nucleotide on DNA. One strand of DNA is opened, as shown in this diagram here. That we know that this DNA is made up of the strands like this. So one of the strand is open here like this, and then mRNA enters into that and it reads the nucleotide adenine, thymine, guanine, cytosine. It reads that, and according to it, it prepares its own codes here like this. Only thing is that instead of thymine in the DNA, there is uracil in RNA. That only one nucleotide is different, and this process is called transcription. Process of RNA synthesis means the process of creating mRNA by reading the codes in the DNA. This process, the process depicted here, this is called transcription. Reading the codes of DNA and transferring the codes into mRNA in the nucleus of the cell. This is called transcription. Only difference is that instead of thymine, RNA contains uracil. mRNA formed in such a way, it comes into cytoplasm. It comes out of the nucleus through nuclear pore and it enters into cytoplasm. It brings the coded message from DNA. It's like a postman. It is carrying the messages which contain the recipes for creating the proteins. The message contains codes for amino acids. The code for each amino acid contains 3 nucleotides, and that is called triplet codon. That is adenine, guanine, uracil, cytosine. Out of these 3 nucleotides, they form a triplet codon which contain information about a particular amino acid. Dr. Hargobind Khurana, he discovered the triplet codons for 20 amino acids, and for his work he was awarded Nobel Prize in 1968. So we have seen that mRNA comes out of the nucleus with thousands of triplet codon on it. Now, according to these triplet codons on mRNA, there is one more RNA called tRNA or transfer RNA. Transfer RNA has a complementary sequence of codon on mRNA, and it prepares itself according to that triplet codons on mRNA. So, formation of anticodons on tRNA as per the triplet codons on mRNA is called translation. That means here the tRNA is bringing the amino acid molecules from various cell parts according to the codons on mRNA. And this tRNA has its own anticodon which have complementary sequence to the codon on mRNA. This process is known as translation. That means bringing the amino acids as per the codons on mRNA by preparing anticodons on tRNA is called translation. First was transcription, where mRNA read the message from the DNA, and here tRNA is fetching the amino acids as per the triplet codons on mRNA by making anticodons complementary to the triplet codons on mRNA. Then these amino acids brought in by tRNA, they are bonded together by peptide bond with the help of rRNA. And during this process, rRNA moves from one end of mRNA to the other end of mRNA by the distance of one triplet codon each time, and this is known as translocation. So rRNA is moving through mRNA, it is browsing through mRNA, it is going from one end of mRNA to the other end of mRNA, and it is bonding the amino acid brought by tRNA using peptide bonds and preparing the proteins, complex proteins, which are necessary for the life processes of that cell and of that organism. So we have seen three main processes. First, this entire thing, central dogma we have seen. So central dogma is synthesis of the protein by DNA through RNA. And in this central dogma, there are three main processes, mainly transcription, then translation and translocation. First is transcription. What is transcription? Transcription is making of mRNA, correct? Means sequence of nucleotide in mRNA is produced as per the codes on the DNA, and this process is known as transcription. Then there is tRNA, which supplies the amino acid as per the triplet codons on mRNA by forming the anticodon, which are complementary sequence to mRNA. And finally, the translocation process, which is motion of rRNA from one end of mRNA to the other end, reading the triplet codons and then forming the peptide bonds between the amino acid brought by the tRNA, and finally forming the complex proteins. So this is the entire process taking place in living thing. This is life, you can say. Means life is all about making complex protein, and this complex protein are made by the process of central dogma, which contain three main processes, namely transcription, translation and translocation. So these processes take place in the living thing, and living thing, they transfer the characters to the new individuals like themselves by the transfer of genes, the transfer of chromosomes. So living organism can produce new individual like themselves due to genes only, and some of the genes are transmitted to the next generation without any changes, and due to this some of the characters of parents are transmitted to the offspring. So the question can be asked, how the characters are transferred from one generation to the other. The answer lies here. They transfer the chromosomes to the next generation, and this chromosome contain the information about the proteins which decide the structure and function of that organism, and hence the new generation, they carry all these characters. Sometimes what happens, there are sudden changes in genes, sudden changes. Now these sudden changes can occur by replacement or by means skipping a particular triplet codon or by any other way. So that is called mutations. Sudden changes in the genes are mutations. Any nucleotide of gene changes its position and that causes a minor change, that is called mutation. Some mutations can be minor, but some can be serious. For example, in sickle cell anemia, the mutation is such that the shape of the red blood cells change and that causes anemia. Anemia is insufficient supply of oxygen to the cells. This is an everlasting process, and it is one of the proof of Darwin's theory of natural selection. So this mutation, they can produce some genetic disorders which is not good, but some of the changes can be favorable to those organisms and they can help in natural selection. So mutation is one of the proof